Thank you, Rob, and thank you to ARM and the organizers and sponsors of the meeting. It's been a great meeting so far, and I hope we all get a chance to interact throughout the next several days. So, forward-looking statements. We are a publicly traded company. Agtc has really diversified its portfolio over the last year and a half, and we're creating significant value for a wide range of patients and stakeholders. Today, I'm going to be focusing this conversation on the data that we released last week in 3 of our clinical trials. And it has really validated our platform, shown that we have the expertise both in the science and the manufacturing and in the clinic to bring products forward that are going to make a difference in patients' lives. And this early evidence of biological activity and efficacy in these clinical programs really strengthens our resolve to continue bringing products forward for patients in a wide variety of areas, both in ophthalmology, in otology, and in CNS disorders. We have a core value of keeping our patients in mind, and we have long had very strong relationships with the patient advocacy groups within ophthalmology, not just the Foundation Fighting Blindness, which is one of the seminal groups in the area, but all of the very patient-specific, indication-specific groups across the country. And this is really critical to development in rare disease because many times you learn from the patients things that are very important to clinical development, such as in Achromatopsia, we learned that really the most important symptom for them was light sensitivity, and we've built that measure of light sensitivity into our clinical development. So understanding the patient, what the journey they take, what is important to them in their lives, what it is they're looking for in product development is very important, and we have kept it in mind from the beginning. So we do have a strong balance sheet. We have over 1.5 years of cash on hand, which is more than enough funding and resources for us to continue to develop the programs. Today, I am going to focus on the first 3 indications on this product pipeline slide in the following slides: X-linked retinitis pigmentosa and 2 different Achromatopsia trials. But I do want to point out that we have other programs in ophthalmology. We have a partnership with Bionic Sight in New York City and optogenetics. We recently announced a collaboration with a local company here in San Diego, Autonomy, where we're going to be developing a product for one of the most prevalent causes of genetic causes of hearing loss. So it's a new indication area for us. And then we have several programs in CNS. Importantly, one of our newer ophthalmology indications is in dry AMD. So this is really going to transform us and move us forward from single focus in orphan indications into larger market indications. So now I'm really going to hone in on the 3 clinical trials that we have ongoing, the X-linked retinitis pigmentosa and the 2 Achromatopsia trials, and the very exciting data that we released late last week. So X-linked retinitis pigmentosa is one of the many forms of retinitis pigmentosa. They're missing a protein that helps transport other proteins and complete the phototransduction cascade. It's about 20,000 patients in the US and EU. And it hits very early in life where they get night blindness and then constriction of the visual fields until between 45 and 55, these patients become completely blind. So it's a very devastating disease to the patients. It's something that they notice and recognize each and every day, that their vision is getting poorer and poorer. And we are very pleased to announce the results I'm about to present. There we go. So we had a 2-part trial in XLRP. We were dosing patients peripherally, either peripherally or centrally, because as I said, this is a disease that starts in the periphery and constricts into the central vision. And we really wanted to explore where was the best place to treat, where could we have a good effect. And so the first piece of information we were able to say is that for patients dosed peripherally, of which there were 9 in this trial, we were able to show that especially at the middle and high doses, we were stabilizing their vision. Remember, this is a disease where the patients get worse and worse and worse every day. So the eyes that were treated are the red lines, And you can see in that middle and high dose, the vision is stabilized compared to the untreated eyes that are in gray. Now, it's very early in a lifetime degenerative disease to really fully claim stability, but we think this 6-month data really points us in the right direction. Now, the other place that we treated patients was centrally. So patients with XLRP preserve a good amount of their central vision for many decades of their life. But as I said, in their 40s and 50s, even that goes away. And we were very, very pleased to see that in the centrally treated patients, we actually saw an improvement in their visual fields. And this is shown here with the red lines showing an improvement in the decibels. So decibels is a measure of light, and so we saw these patients improve their decibels. Here you'll see a slight decline at month 3. One patient had to have a surgery that was not study drug related that caused a decline in visual fields. We fully expect that at month 6 that decibel level will go back up. On this slide, we have compared the data to other reported data by our peers, and you can see that using the exact same measure, mean sensitivity, mean improvement in visual sensitivity, that we're very comparable and a little bit higher and a little bit more robust signal there. Then, if you look at a different way of looking at, no pun intended, visualizing mean sensitivity, you can look at it in a heat map function. So what this is doing is plotting on the eye the area that the patient has visual function in. And so the idea here is that you'd like that colored area to be as big as possible, or you would like the color within that area to get brighter and brighter, because the brighter colors represent more sensitivity. So as you can see, the treated eye is up here on the top, and the area is getting bigger over time, and the brightness, this yellow area, is getting brighter and brighter, compared to the untreated eye, where there's no change in that tone of color, and there's really no discernible or statistically meaningful change in the area. So this is a representative sample of the responders in the trial. We had another patient that looks very similar to this with an expanded area and a brighter area, and then we had 2 patients that had some sensitivity over a larger area, but it got much brighter. So we're very pleased that both this way of thinking about sensitivity as well as that mean sensitivity plotted on the line I showed you all took together show that these patients had a change and an improvement in their vision. And then the last way we can think about and look at the patients in the trial that responded centrally and saw improvements is with visual acuity. So I think most people are familiar with visual acuity. It's how many letters you can read on an eye chart. And what you can see here is that the average increase in the treated eye was about 5 letters. And we actually had several patients that had as much as a 2-line improvement or 10-letter improvement in visual acuity, which is really quite significant for this patient population. And that was compared to just 2 letters and no patients that had really that dramatic 2-line improvement. So here's 2 ways to look at visual sensitivity and visual acuity that taken together say that in centrally treated patients, we were seeing an improvement in visual function at the 3-month time point, and this was really an early time point for us because our standard time point to measure is 6 months. So we're very much looking forward to the 6-month data that we'll be releasing by the end of this year. And so the conclusions here are that the study drug was safe and well tolerated, that peripherally dosed patients were experiencing a stability in their vision, which is really life-changing for these patients, and that the centrally treated patients were seeing an improvement in their visual function. So our next steps are that we'll be releasing the 6-month data by the end of this year. We'll be following up with the 12-month data in the first part of 2020. We're going to be having ongoing discussions with the FDA to come to agreement on our pivotal trial design and expect to be starting that pivotal trial in 2020. Achromatopsia is another orphan inherited disease, about 28,000 patients in the US and EU, but then all of the similarities to XLRP cease because it's a very different disease. It's a cone-only disease. They have cones in their eyes, but the cones don't work. So what this means is that they're extremely light sensitive or have discomfort in lit conditions. This would be an impossible room for them to be in. And even in the best of lighting conditions, which are very dim, their vision is still less than 2200. They're all legally blind, and they have absolutely no color vision, black and white and shades of gray. So it's a very limiting disease. We're presenting very early preliminary data in achromatopsia because we only have 3-month data on an endpoint that is a very unique and powerful endpoint that we developed with our collaborators at Bascom Palmer, and it has to do with that light discomfort. Because the patient said this was the most important thing to them, we thought it was important to develop a way to measure this and report back to the patients on the thing that was most important to them. So this is a quantitative way to measure the maximum light that a person can tolerate. I can tell you here in this room, this is about a log lux level of 3 or 4. It's quite bright, and the patients would not be able to tolerate it. Most achromatopsia patients prefer it to be about 0.5 to 1 log lux, which would be like a dark starry night. And what we were excited to see in the Achromatopsia B3 trial is that we saw what looked like a dose-dependent response of getting a clinically meaningful improvement in that log lux level. So in the mid-dose, we had 1 out of 3 patients see a very significant improvement in their treated eye, and in the high-dose group, we saw 2 out of 3 patients. So we're going to look very much forward to the 6-month data by the end of this year, where we'll look not just at this light discomfort test, but then we'll be able to pull down visual fields, color vision testing, visual acuity, and put all that data together. Similarly, in the A3 trial, which is the kind of sister trial to the Achromatopsia B3 trial, again, at that mid-dose level, we saw 1 out of 3 patients get a response. We don't have the high-dose patients yet. But we're very excited by this data in achromatopsia on the most important endpoint and look forward to collecting the rest of the data to move forward. And then these 2 trials would again be able to move forward into pivotal studies. So I do just want to take the last couple of minutes of the presentation to talk about the background of the company and what supports these clinical trials. First and foremost is that we're a very technically savvy company. We have been operating in this space for a number of years. We carefully look at the design and construct of all of our vectors in non-human primates. We select the capsid, the promoter, analyze the gene cassette in non-human primates because primate eyes are very different than mammals. And if you're doing your early work, your preclinical work in mice or dogs or sheep, you're not going to know if you're targeting to the right place in a primate eye because the structure is quite different. And I think that's a very important aspect. And then when it comes to manufacturing, I know that many people say in ophthalmology the doses are so low, you don't really need to worry about manufacturing. Well, I think that many of the news articles that we've been reading over the last year or so, where there's been missteps in manufacturing and the FDA is really focusing on that CMC section, tell us that manufacturing is very important. And this is another area of excellence for us. We've been working on our manufacturing system for over 10 years. It's been in 7 INDs. It's been reviewed by the FDA, the Israeli Ministry of Health, the Canadian Health Authority, and the Irish Medicines Board. So we really have the experience interacting with regulators, having not just our process under control, but our assays fully developed and having a complete and full C of A. And I would like to point out that we have in our current system, we produce over 4,000 doses in 25 liters. These are single-use bioreactors, serum-free, in suspension. And as I said, that's a lot of doses because it's ophthalmology, but even in ophthalmology, there are people that are taking 100 liters to get about 100 doses. So this is a very significant productivity advantage that we have. This is our team. I think you'll see from the team that we have a lot of experience in cell therapy and gene therapy and ophthalmology. Our most recent hire was Teresa He, our new Chief Medical Officer from Erie. She has tons of experience in late-stage development and commercialization, which is exactly where we're heading, and that was an important add for us. And here's our pipeline again. The message is 3 sets of positive clinical data in our 3 clinical programs, a very rich pipeline that adds to the ophthalmology portfolio, but also expands us into CNS and otology, and a cash runway that will allow us to complete these trials and move into the pivotal trials in 2020. So thank you for your time and attention, and I hope you have a good rest of your conference.